Hello, my name is Alan Moshak, and I'm a research instructor at the University of Utah Metabolomics Core, pictured here in the center with the beard. And this video has been created to familiarize collaborating researchers into what lipidomics is and how we here at the U operate our lipidomics workflows. Be sure to also watch two videos on metabolomics and sample submissions that our director James Cox has created. Why should we study lipidomics versus the other established fields like genomics, transcriptomics, and proteomics? Well, lipids are classified as metabolites, and along with other small molecules such as sugars, nucleotides, and amino acids, they are the closest that we have to the cellular phenotype, which tell us what is happening and what has happened within the cell, versus, say, the genotype, which is really just a blueprint. Lipids are a very diverse group of naturally occurring molecules that play a variety of critical cellular functions. They constitute cell membranes, serve as a matrix for transmembrane protein function, energy storage or homeostasis, play independent roles as intra and intercellular signaling, or become precursors to other metabolites. The disruption of lipid metabolism pathways are known to be associated with the onset and progression of various metabolically linked diseases, including obesity, cancer, diabetes, and neurodegeneration. How are lipids structurally defined? A lipid is generally considered to be any molecule that is insoluble in water and soluble in organic solvents. Biological lipids usually refer to a broad grouping of naturally occurring molecules, which include anything from fatty acids to phospholipids, plus fat-soluble vitamins such as vitamin A, D, E, and K. Lipids may be broadly defined as hydrophobic or amphipathic small molecules that originate entirely or in part by carbanion-based condensations of thioesters and/or by carbocation-based condensations of isoprene units. Using this approach, lipids have been divided into 8 categories. The most widely used classification system has been established by the Lipid Metabolites and Pathways Strategy Consortium, also known as lipid maps. They divide lipids into 8 classes based on their functional backbone: fatty acyls, glycerolipids like di and triglycerides, glycerophospholipids like phosphatidylcholines, sphingolipids, which include ceramides and sphingomyelins, sterol lipids, prenol lipids, saccharolipids, and polyketides. What is lipidomics? Simply put, lipidomics is the quantitation of the lipidome. As a pure workflow, lipidomics consists of sample preparation, sample purification, sample analysis, and data analysis. More specifically, lipidomics is a systems level analysis of lipids found within a tissue, biological fluid, organism, culture, or other biological source, falling under the umbrella of the larger field of metabolomics. Most often we examine the lipid differences found between biological samples after perturbation of a given system. Some of the questions that lipidomics asks are: what lipids are present and how much? Where do they originate from? Are they interrelated? Are they normal or are they abnormal? What are their functions and are they associated with disease? While there is no single analytical platform that can fully characterize the highly diverse lipidome, our sample extraction and preparation strategy, coupled with reverse phase UPLC-MS, provides a robust tool to assess the lipid content of any biological system. Our lab primarily uses the MATYASH lipid extraction protocol that employs methyl tert butyl ether, or MTBE, and methanol, and along with water, to execute an efficient liquid-liquid extraction for broad coverage of the lipidome. The extraction starts with an incubation of the sample with MTBE, methanol, and internal standards. We rely on a variety of internal standards depending on the needs of the researcher. The premixed internal standard SPLASH lipidomics from Avanti Polar Lipids is our default internal standard. This mix contains 14 deuterated standards representing all of the major lipid classes and works for both targeted and non-targeted lipidomic studies. We then add water to induce phase separation followed by centrifugation, whereby the upper phase containing the lipid fraction is removed and dried down under nitrogen. Using MTBE here, as opposed to chloroform, which the Folk and Bly-Dyer methods use, allows us to avoid having to transfer from the bottom layer. It lowers costs, improves stability, and has less toxicity compared to chloroform. That said, we are perfectly willing to utilize those other extraction methods at a researcher's request. It's also important to note here that we have instructed several researchers on this technique so that they can prepare the lipid extracts themselves, which improves the throughput and lowers cost on the researcher's end. Researchers would then only pay for instrument and analysis time when they prepare their own samples. We use two different sample types for quality controls. The first is the System Suitability Test, or SST, that is a pre-prepared commercial extract from Avanti Polar Lipids. We use a freshly thawed aliquot before each batch to monitor instrument performance. The second QC sample type is a pooled quality control sample that consists of small aliquots from each sample or sample group. We also use these pooled QC samples to build lipid annotations and databases, which I will discuss later. There are two different types of blank samples. The blank is also called an extraction or process blank and consists of everything seen during the extraction except for actual biological material. This allows us to monitor any artifacts picked up during the extraction or run and eliminate false positive lipid annotations. We also use a double blank, which contains only the resuspension buffer. We have two workflows for lipidomics. A non-targeted catch-all method that utilizes an Agilent 6530 UPLC QTOF that scans mass ranges from 90 to 1700 Daltons. This is typically the workflow of choice when researchers are in a discovery phase of a project and require broad screening. It offers relative quantitative analysis, very high mass accuracy, detects and annotates 100 to 1500 lipids depending on the source material and level of confidence required for annotation. Runtimes are longer than the triple quad, and it has a smaller dynamic range. Conversely, the targeted workflow utilizes an Agilent 6490 UPLC triple quad that requires all lipid targets to be known and input into the method before data collection. We use this when we know exactly what targets we're looking for, and also when a wide dynamic range and/or increased sensitivity is required. Triple quad methods can detect anywhere from 1 to over 400 lipids, though in practice, we typically cover about 150 lipids. Now some background on the two different mass spectrometers. The quadrupole mass filter, or QUAD for short, was invented a half century ago by Nobel laureate Wolfgang Paul, and has become an integral part of some of the most sophisticated mass spectrometers. Consisting of 4 parallel metal rods on which a fixed direct current voltage and alternating radio frequency voltage are applied, quads allow users to selectively filter all ions except those of a specified mass to charge ratio, noted as M/Z, depending on the applied voltage. QTOF instruments pair the quadrupole and collision cell with a time-of-flight analyzer to determine an ion's mass-to-charge ratio. Ions are accelerated by an electric field of known strength. This acceleration results in an ion having the same kinetic energy as any other ion that has the same charge. The velocity of the ion depends on the mass-to-charge ratio, with heavier ions of the same charge reaching lower speeds. The time that it then takes for the ion to reach a detector at a known distance is measured. This time will depend on the velocity of the ion, and therefore is a measure of its mass-to-charge ratio. From this ratio and known experimental parameters, one can identify the ion, allowing high-resolution, high-mass accuracy analysis of all ions simultaneously. For example, I've included this spectrum from an MSMS or MS2 experiment that gives us both the mass of the intact molecule along with diagnostic fragments like the acyl chains, allowing us to annotate the lipids down to a very high level. Let's take this moment to discuss lipid nomenclature and what can and cannot be described with the instrumentation we have in our facility. Community accepted guidelines for lipid annotation have been implemented by lipid maps and are meant to completely characterize the lipid molecule as shown here. However, conventional LC-MS or tandem mass spec experiments cannot be used to generate all structural information of a given lipid molecule. Therefore, shorthand notation has been proposed to only confer the level of structural detail known based on the experimental data. With accurate BAS and some class-specific fragmentation data, for example, the loss of the choline head group, we can annotate the sum composition where the class, total carbons, and total double bonds in the acyl chains are known. With additional fragmentation data beyond the head group, we can start to assign the acyl chains. But whether those chains belong on the SN1 or SN2 position can rarely be definitively confirmed with the instrumentation we have at the moment. To that end, an underscore between the acyl chains is used to imply that the acyl chains are not specifically assigned. Changing gears now to the triple quadrupole mass spectrometer. As with the QTOF, the quad initially filters the selected ions, or precursor ions, before reaching a collision cell where they are fragmented into product ions. The collision cell is typically called the second quadrupole, although it's not technically a quadrupole. The fragment ions formed in the collision cell are then sent to the third quadrupole for a second filtering stage to enable a user to isolate and examine multiple precursor to product ion transitions. We call these MRMs, which stands for multiple reaction monitoring. I've illustrated this here with ceramide 18,1,16,0. The precursor ion is the intact molecule with an M/Z of 538.5. During a predefined time segment, other masses are filtered out of the first quad. In the collision cell, fragmentation occurs whereby the amide bond of the ceramide is broken. The compound rearranges in a predictable fashion, resulting in the characteristic sphingosine fragment with an M/Z of 264.3. Other masses are filtered out here as well, and it's this filtering that leads to such excellent signal-to-noise and sensitivity values. The instrument allows us to monitor up to 200 MRMs at once over a defined retention time window. We typically use 2 MRM transitions per molecule over a 10-minute window. So a 10-minute run could theoretically collect data on 1,000 compounds, but of course it doesn't work that easily in practice. For example, our routine targeted sphingolipids methods, which includes a subset of phospholipids and glycerolipids, has over 150 total lipid targets and could easily be expanded if a researcher required it. Let me give you an overview of our non-targeted workflow once the samples have been extracted. Resuspended lipids are injected using an autosampler and separated on a C18 column maintained at 65 degrees that is connected to our QTOF. Samples are randomized and injected twice, once in positive mode and once in negative mode. Each injection takes 23 minutes, including equilibration. The pooled QC samples are subjected to LC-MS using iterative exclusion to build a fragmentation library, and a subsequent lipid database is pulled from the Agilent Lipid MS and Silico database and queried against our data. An average experiment annotates approximately 300 lipids at the acyl chain level. We can also use accurate mass on the MS1 level to search the lipid maps database and further annotate lipids at the sum composition level, but note that this will introduce some false positives without the requisite accompanying lipid class fragmentation. Once those databases are created, we integrate the raw data to generate data tables. From there, lipid targets are normalized to the spiked internal standards based on the ratio to the internal standards. The data table is exported and evaluated using Excel and Reporterator, where initial lipid targets are parsed based on the pooled QC's %CV, or relative standard deviation, as well as the ratio of the target in the blanks to the pooled QCs. After data is collected, it requires some basic processing steps. Reporterator is an in-house University of Utah metabolomics built software used to perform these processing steps in a fast and reproducible way. Processing often involves checking for compounds' performance and normalizing to quality controls or an internal standard to remove variation due to instrument noise or sample preparation. Reporterator's most common and most important function is using the powerful metaboloanalyst R to perform statistical analyses and generate figures from the results. More on metaboloanalyst in a minute. A very detailed report containing background on lipidomics, abbreviations, experimental, and figures and tables based on the comparisons requested by the researcher is automatically generated. Our default statistical analysis includes PCA and volcano plots. From these analyses, outlying samples can be identified and compounds with large differences between experimental groups can be found. Additional analyses are available, of course, so please let us know what you might have in mind. Reporterator allows fast and reproducible data analysis, so more time can be given to the unique aspects of each experiment. As I mentioned at the top of the presentation, our director, James Cox, has made a video that goes into more detail here about using Reporterator, metaboloanalyst, and statistical analyses associated with those programs. There are a few software-based tools beyond Reporterator that you can use to help make sense of the data. Metaboloanalyst is a free software that allows for statistical, functional, and integrative analysis of metabolomics data. It is designed to address two common types of problems. One, to identify features that are significantly different between conditions, like biomarker discovery, and two, to use the metabolomic data to predict the conditions under study. In addition, metaboloanalysts can also provide tools for compound identification and pathway mapping for annotating significant features. Note that metaboloanalyst is designed for analyzing large numbers of samples. Very few samples, like less than 10, will cause some functions to work improperly. Users receive data tables formatted for use in metaboloanalyst, as well as instructions on how to get started. Additional analysis is also done using the LionWeb lipid ontology software platform. The Lipid Ontology Enrichment Analysis web application is a novel bioinformatics tool for lipidomics that enables users to search for enriched lion terms in lipidomic subsets. In other words, in your CSV files that we'll give you. Lion terms contain detailed lipid classification from lipid maps, as well as biophysical data, lipid function, and organelle associations. BioPan, or Bioinformatics Methodology for Pathway Analysis, is a tool that allows users to upload their own lipidomics dataset and perform a pathway analysis. In this analysis, you can also explore systematic changes in lipid pathways at different levels, the lipid class and lipid species. The researchers at BioPan have created a very helpful YouTube video tutorial, so please give that a look. Metaboloanalyst, BioPan, and the Lion software all require uniquely formatted CSV data tables that we provide to all researchers. We ask that researchers collect and include the following background information when submitting samples. James Cox has created a separate video to lead users through an online sample submission process. That submission is typically accompanied by informal chat or email exchange about the background and goals of an experiment. So please reach out with any question, big or small, at any time. Sample collection is a vital part of lipid analysis. With the highly sensitive analysis methods available, only a very small amount of source material is needed. 2 points should be kept in mind. 1, the small quantity of sample taken should be representative of the entire source material. And 2, just enough material, but not too much, should be sampled for all of the planned analyses. What is enough material? There is no clear answer for this, as all source material will vary. Generally, we recommend a minimum of 5 milligrams of wet tissue, 1 million of cells, 5 microliters of plasma, or 100 micrograms of protein from a membrane fraction. We also require researchers to record those values when preparing those samples. With few exceptions, all samples should be flash frozen, stored under nitrogen if possible, and shipped on dry ice. Researchers are free to label their samples as they see fit, although we strongly recommend a simple labeling scheme that refers back to detailed sample information. If possible, researchers should generate a blank or negative control sample in their set. We also request that researchers ship their pre-weighed tissues in the bead mills tubes shown here. And in summary, what can you expect when you take on a lipidomics project with the course here at Utah? You can have guidance on experimental design if you need. Of course, we take the samples through our workflow of sample preparation and extraction, and sample purification and analysis on our LCMS platforms, and finally data analysis using our reporterator. We can also instruct you on how to prepare and extract your samples yourself if so desired. You'll get a Word document with the background and methods, tables and figures, Excel file with raw and normalized data tables that are ready for use in MetaboAnalyst, Lion, and Biopan. You also get statistical analyses and figures from MetaboAnalyst, Lion, and Biopan. You'll also get a report summary highlighting significant differences between the experimental groups. And finally, turnaround times can vary widely depending on just how busy we are at the core. Approximately 2 to 3 months from sample submission is pretty close to normal. That comes to the end of my presentation. Thanks for listening. Like I said, please feel free to reach out anytime with any questions or comments.